Recording now. Okay, so welcome everybody again with this June seminar of our series for the DRIVE Health Doctoral Center at King's. We are expanding our audience a little bit also to other researchers and colleagues from the NHS trusts that collaborates with us, and so whenever you receive an invitation from me for these seminars, feel free to share within King's and within the collaboration at NHS that you may have. Today we have a guest from UCL, so very close to us. Dr. Langdon is currently serving at UCL in the Software Systems Engineering Group. We first got in touch through the genetic programming bibliography that he's maintaining on his website. It indexes more than 15,000 papers on genetic programming, and that's why we met. I was recently added to his database because of my paper on symbolic regression. So he's an authority in his field. He's author of 3 books on genetic programming, co-organizer of many editions of GECO, the leading international conference of the topic, and winner of international awards and fellowships. He was kind enough to accept my invitation to speak to you today. So we will hear about his experience in genetic improvement of software. As always, I'm recording these seminars, so if you're not happy to appear in the video, this is the moment to leave. Yeah, we will have a presentation, a Q&A session at the end, and the stage is yours, Professor.
Right, so what I'm going to try and do now is share my screen. So hopefully at this point you should now be able to see the first slide.
Yes, we can.
Okay, so I'm Bill Langdon. I'm at University College London. I made rather too many slides, so I'm going to skip past various bits, but if you see stuff flash by and you want more information, then let me know. So as I say, it's Bill Langdon, and the topic is genetic improvement of software. And essentially the idea with genetic improvement is we want to use the power of evolution within the computer, applying evolution to programs. I'm going to talk a little about some applications that we've been doing, mostly regarding speed ups, but also other areas. What I want as a general takeaway is people to take home the idea that software is plastic, it's robust, and you can evolve it. So firstly, what is genetic improvement? And essentially the idea of genetic improvement is we're going to use evolution on existing programs. So it differs from an earlier technique known as GP, where the programs were created from scratch each time. Here we're optimizing code that already exists. And essentially we take the ideas of Darwin. We have a population of programs inside the computer. We assign fitness to them. So the fitness is essentially gathered by running them. So do they produce the right answers? Do they run quickly? Do they run in a small memory footprint? And then we select those that are better according to our fitness measure to be parents for the next generation. The next generation is created typically by a combination of mutating the parents and also crossing over the parents. So in typical form, we would start on the left-hand side with manually written source code. That's automatically converted into some internal representation, such as a grammar or an AST, which is the typical representation used within compilers. And then we have a population, not of programs, but of changes to the program. So we don't have to store many copies of the program. We just have copies of changes, to typically much smaller. We operate always on the changes themselves. So we have specialized mutation operations for changing those patches, for crossing over patches. We then effectively reverse our grammar to produce program that we want to test. Assuming we can compile it, we try and run it on test cases, and then we select the better programs to be parents of the next generation. And then we go around this loop maybe 10 times, maybe 100 times, produce a list of changes that we want to make the program, and typically there's also an automatic cleanup process, so we just include the changes that are necessary. So the idea really is genetic improvement is starting from the existing system. It's starting from non-random code. So not only do we have the existing system, but we can use parts of that existing system as a source for new changes. We also use it as essentially as a test oracle. So we can always compare our new version of the program against the old one. And typically this is much easier way of saying whether the program is good, rather than trying to generate a watertight specification. So typically we can use testing, we can produce variants and test them, and that becomes our fitness measure. In terms of genetic improvement, the big success would be in terms of automatic bug fixing, also known as automatic program repair. And this is where things have really changed quite dramatically. So in the last 15 years or so, I mean, 15 years ago, the idea of having a computer which automatically fixed bugs in its own programs was totally ludicrous. Fortunately, the inventors of APR did not take that to heart. They were not told that this was impossible, and they just went ahead and did it. And they've received multiple prizes, best paper awards. One particular example that highlighted in blue, again, the work of a PhD student. This is Sammy Harrelson in Stirling. He was involved with creating Python code for an Icelandic health clinic. This is the Janus management software. And the typical way that system is used is users come in at about 9 o'clock, use it intensively during the day, and the last person logs off before 6. In the development, if there are bugs discovered, then the system logs those bugs. And when the last person leaves the office, Sammy's automatic genetic improvement system kicks in and starts looking for fixes for those bugs. It runs overnight, emails the fix to the developer by 8 in the morning, and then the developer decides whether they're going to use the fix or not. And in the first 6 months of operation, they got 100% record. They managed to fix all of the bugs. The GI fixed all of the bugs automatically, and they were all accepted by the development team. So this system is still in use. Over the course of the next 2 years, they lost their 100% record, but nonetheless, it's an impressive number of fixes that have been automatically generated. Facebook now have their own system, which is similar idea, which is being used on a truly industrial scale. Again, generating fixes on a pretty much a daily basis. The other big application of genetic improvement has been to speed up code. So I'm going to talk particularly about the Bowtie 2 example from bioinformatics, where we got programs which were 70 times faster on the particular thing they were trained to do. Barracuda is another bioinformatics application. This was the first time that genetic improvement was actually used. It's been available. The genetic improvement version has been available since 2015. More than 7,000 downloads. It was used by IBM in one of their supercomputers. It's been used on the web, and more recently for epigenetic work. And again, RNA fold, I'll talk a bit more about that, but it's a bioinformatics tool looking at predicting the structure of RNA. Other examples are using genetic improvement to reduce the energy consumption, particularly of mobile phones, reducing memory footprint, reducing runtime. Another example is particularly with mature languages like Java. There are often many libraries that have similar functionalities, and what Jerry and his colleagues were able to show was that you could use genetic improvement to choose the most appropriate library for your particular application. And indeed, we've shown that not only can you improve programs, but you can also use GI to improve the libraries themselves. Another example, so this is actually from Alex, who is a PhD student in UCL. Again, nobody told him this was impossible. So what he did was to take an open source editor, Kate. So Kate is a standard library, but the sources are available, and show that you could use genetic improvement to automatically include a source from elsewhere. So his particular example, well, several examples, but this particular example is the H.264 codec, which he was able to show that you could transplant that into Kate. The runtime for the GI system was 26 hours versus an estimate of 22 days to do it by hand. Also looked at things like porting code in particular to parallel hardware, GPUs, graphic cards within systems, and parallel vector instructions, the SSEs. So that another example from a PhD student. This is Pichera, who was in University of Washington, and what he showed was it was possible to adapt graphics cards shaders. So these are notoriously difficult pieces of software to write. On the left, we have 3 different renderings of a marble dragon. So the problem there is if you spend a long time rendering or writing the code to render, i.e. to put on the screen a particular model such as this dragon, and then the boss says, oh yeah, but we need 100 of these dragons. We need to do this in real time. How can you adapt your code to generate 100 of these things rather than just one? And the answer is you can't put 100 programmers on it. But what you can do, and on the right-hand side, you'll see each of those dots represents a variant of the program automatically generated. And what the dotted blue line is showing is a Pareto trade-off. So if you take the point labeled A, you see it's actually quite a lot faster than the original code, but it's slightly more makes slightly bigger errors. In the panel, the point B is shown, so that's considerably faster and a small number of errors. And you can the point being that you can automatically change the software to do something slightly similar to what it was doing before, but possibly a lot more efficiently. And again, in principle, you can do this by hand, but it's very tedious, and you don't know in advance how much effort you're going to need to achieve a particular goal in terms of the speed. I'm going to skip past that one just to show, just to say the RNA fold has been downloaded a huge number of times, and indeed it's incorporated in the online server, which has been used more than 1 million times. This comes from Justina Pecte's survey. And what I wanted to stress there really was the dominance of the amount of work that's been done on automatic program repair, which is in red, and on runtime minimization, which is in green. So you can see those are the two popular ones. But there's a lot of space for new ideas. So it's by no means exhausted what can be done. So as promised, I'm going to talk a little bit in detail about Bowtie 2. So it's bioinformatics application. It was very much the right program at the right time. When B.Langdon released it as part of his PhD, the human genome had just been sequenced and published. There was a huge volume of next generation DNA sequences being generated, which had to be aligned against the reference genome. Bowtie 2 was the tool that made this possible to do this on a fairly upbeat laptop, but on a general machine rather than on a node in a supercomputer. And as I say, it's very much the right program at the right time. Within a year of releasing it, it had attracted more than 1,000 citations. What we're going to do is take that existing C++ code, use that as the oracle of what should be happening, and we're going to use GI to make changes to the code and train it to, in particular for one task, using data from the 1,000 Genomes project. So it's a non-trivial piece of software, 50,000 lines of C++. And as I say, what we want to do is target short sequences generated by the 1,000 Genome project. As before, our approach is to convert the code into a grammar and then manipulate the grammar. So we're making small changes. The grammar guarantees that the new code is syntactically correct. Doesn't guarantee that it's going to compile, but it will at least be syntactically correct. In fact, and this is generally true through GI work, including program repair, bug fixing, most of the time when things fail to compile, it's simply because variables have been moved out of scope. Here, 3/4 of the mutants compile, and of those that compile, most run okay. Most run in the sense of running without error and generating a plausible answer. So going back to our evolution picture, our approach here is to take the code, automatically convert it into a grammar, and then make small changes to that grammar. That becomes a population of patches. We can manipulate those patches. We can add them together. We can change them, give rise to a new population of patches, and we effectively invert the grammar to get a new variant of the program, which we then attempt to compile. As I said, most of the time they do compile, and then we run them on test cases from the 1000 Genome Project. We select the top half of the population to be parents for the next generation. Each of those parents gets 2 children, and the children are created by crossing over between different parents and by mutation. And in this example, we have a very small population, only 10, and we ran for 200 generations, which took 25 hours. So I've talked briefly about that. Essentially, the idea is we're going to use profiling to focus the search. So although we start off with 50,000 lines of C++, more than 100 files, we start narrowing it down by excluding the header files, by excluding code which is not covered by conditional compilation. We have a weighting scheme to put the effort on lines of code that are used most heavily. And as a result of that, of our 200 generations, we come up with mutations which affect 39 lines of code in 6 different files. And by our automatic cleanup, we get that down to seven changes across 3 files. And as I said, this is the speed up it gives a factor of more than 70. So the runtime went from more than 12 days to less than 4 hours on the thing it was trained to do. In terms of the quality of the output, 89% of the answers are absolutely identical. 9% are slightly better. In terms of the quality, there's a small fraction which are the quality is the same, and there's 1.5% where there's a very small degradation in quality. And again, on the particular task it was trained to do. And since then, we've looked at Barracuda, which is a similar idea, but working much faster by exploiting the power of a graphics card, a GPU. So these are highly parallel pieces of hardware. So something like string matching, which is the guts of what the algorithm does, can be spread across many processors. We're able to show a speed up of about 3-fold on very typical applications. And as I said, Barracuda has been downloaded more than 7,000 times. IBM ported it to one of their supercomputers that they built for the US Department of Energy. And essentially, Barracuda is being shown to run on hardware that costs graphics cards that cost £50 up to, as I say, this $300 million supercomputer. Then the last bioinformatics applications are an RNA-fold. Again, looking at exploiting parallel hardware. So this particular example is the AVX vector instruction set, common in many top-end Intel machines. Again, just taking the evolutionary cycle, having a population of changes, and testing those changes on representative data. Again, we're fortunate, and this code has been taken on board by the owners of RNA-fold. It's been incorporated in the Vienna RNA package. It's been downloaded. Our version with the GI changes has been downloaded many tens of thousands of times. As I said, the server in Vienna has been used more than 1 million times. And now it's been out for a while, we're starting to see citations of RNA-fold, which actually point directly at the versions that we know have the GI code in it. So this particular one by Andrews from 2 years ago showed that they were using the updated RNA fold on their work with predicting the structure of RNA used in designing probes for COVID. The last bit, essentially some theoretical justification. So if you bear with me, so this is a large, highly nested function. Each of the components is quite small. It's just an addition or a multiplication or essentially one of the 4 standard operations. I'm going to try and zoom in. The idea is we've taken the original tree and then down here in this red subtree, that's been inserted into the original tree, and then this colored line is showing the disruption to the original code by making this change. It's actually produced by crossover, but you get the similar behavior if you make a mutation. So bear with me if I can zoom a bit in on that. Oops. Okay, so the red code down at the bottom, we drop that in, replacing what was there before, and immediately in the black node, immediately above the change, what that means is the darker the color is, the bigger the disruption, the bigger the change from the original tree. And the number 48 indicates on all 48 test cases, the new code produced a different answer to the old one. As we move up the tree, so the blue indicates that there is a difference on all 48 tests, but it's not as big as the first time. And if we proceed up, 40, we get 48, 48 again, and then we get 47. So what that means is on one test case, the original tree and the new mutated tree are producing absolutely identical answers on one of our 48 test cases. Because it's identical on that test case, it's going, and the system is entirely hierarchical, it's entirely nested, that on that test case, it will always remain identical. So the next node up, we also get 47. As we proceed up, we have this one test case where they're producing identical answers, and then we get one with 38. So suddenly, we now have 10 test cases which are producing exactly the same answer in the original code and in the mutated one. Again, there's a change of color indicating on average the two have become closer, and on 10 of those cases they become identical. So there's another example here where we go from 38 to 37, and you can see the yellow indicating that the difference between the two trees, the evaluation of the two trees, is now becoming quite small. And we go from 37 down to a few, and then there's there are several levels of nestedness where the colors get gradually more and more faint, and eventually there's only one test case left. And in this case, if we go above there, we get to a node where the original tree and the mutated tree, the crossed over tree, produce identical answers on all 48 test cases. So in terms of information theory, this is being called failed disruption propagation. And the idea is, in information theory, all functions lose information. So if you think of an addition, if the result of an addition is 3, then we don't know what the inputs were. They could have been 1 and 2, they could have been 0 and 3, it could be -1 and 4. We don't know what the inputs are from the output. And this is entirely typical, so it's not particularly that example that I've just shown you. All hierarchical nested structures lose information. One of the things that's well established in software engineering goes under the name of the Pi framework, where the idea is if you have a bug buried in your program, it has no impact unless it's executed. So E for executed. That bug, when it's executed, needs to make a change. So what's called here infection. So for instance, if we have 2 + 2 gives us 4, but it should have been 2 times 4. Well, it actually, in that particular test case, produces the same answer. So there's no infection. So in general, the error needs to make a change to the state, and the change of state needs to propagate to the program's output. And what we've just shown in this large example is, as things get increasingly nested, there's less and less chance of propagating disruption to the output. And in fact, in many examples, we see an exponential decay, which essentially means the inverse of an exponential is, of course, a log function. So the effectiveness of test suites, at least given black box independent random testing, it increases slowly. It increases only as log of the number of tests. Another way of viewing this is, what we want to do is put our test oracle, our check for whether there's an error or not, as close to the error as possible, or we have as many oracles as we can possibly fit into the system. So this is what the left-hand chart is trying to show to us. So this is a log scale, and what it's showing is, if we disrupt, here it's an integer program, but it's the same argument as floating point. If we disrupt our program close to the output, there is more chance of that disruption actually changing the program's answer. Whereas if we make a change deep within the program, so here you can see we're nesting 10, 20, 30 levels deep. If we make a change deep within the program, it's got very little chance of actually changing the output. And on the right-hand side, plotted effectively the depth to which a test suite can look into the program. So if we're, and it's on a normalized scale, so just to show the impact of log. So although 100 tests is more effective than just one, you can see it's not radically more effective. So there's a problem with using testing to scale unless you can get the tests to probe deeply into the program. So the little graphic there shows a scene from Alice through the looking glass. So Alice on the right is talking with the White Queen, and the White Queen has just said something ridiculous in the typical Hodgson fashion. And so Alice retorts, "Oh, but that is impossible." To which the Queen replies, "Oh, but you must believe impossible things. When I was your age, I could believe 6 impossible things before breakfast." And I wanted to take that as to stress that to get impact, one of the easy ways is just to do something that everybody else considers impossible. The idea of automatically fixing programming errors, computer fixing its own errors, totally impossible. Fortunately, Claire was not told by her supervisor that it was impossible, and so she went away and did it. And in terms of GI, what the general message is, software is much more adaptable than you might expect. Yes, a lot of mutations go wrong, but if you're prepared to keep trying, then evolution can work not just in nature, but also within the computer. So just to conclude, what I've talked about as being genetic improvement applied to real-world industrial-sized software, much of it open source, some of it, as in the case of Facebook, is of course proprietary. The GI code has been adapted. It is in use worldwide. In terms of particularly the 2 examples with bug fixing and also RNA fold and Barracuda, those codes have been adapted automatically, but incorporated into the normal software engineering processes have been maintained by people in addition to automatic changes. In terms of testing, obviously is we have this rule that independent black box tests will grow ineffectively very slowly, only as log n. So you could, we found examples where 1,000 test cases is only marginally more effective than having 48 tests. And particularly with deeply nested structures, this is going to make testing hard. And what you want to do is place your test oracle, you'll check that whether there's a bug or not, whether the program is behaving itself as close to the potential errors you can manage. And essentially, the idea is we should lose our fear of manipulating code. It's not fragile. Yes, if you mutate it, something may go wrong. But if you're prepared to try multiple times, if you're prepared to have a population approach, then often the code can be evolved to produce great results. And at that point, I really should stop. So thank you very much.
Thank you. Thank you very much. I found this very interesting. So many points and so many links to other useful resources to see and to explore. So I will start. I have so many questions actually, so I will start for sure asking some of them. If anyone in the chat has other questions, feel free to write them down or to raise your hand so we can talk directly. So I would like to ask you your take on more recent topics, and how do you feel about these genetic approach on the software evolution and software improvement, and what's happening with these new AI tools that promise us to write software, and so to correct what we write using machine learning. So it's a completely new thing, and What's your take on that? Well, how do you see these things evolving in the future?
Well, so I should stress that it's not my area. So,
Okay.
In terms of practicalities, then the systems that I've seen have been built on using vast numbers of human written code, and so they're potentially well, several problems, but I guess the principal problem from our point of view is the size of these systems make it infeasible for most universities to go down that route.
Right.
It's very the systems that I've seen, you cannot hope to train them on current computer science hardware. That maybe that's an argument for saying we should club together and get sufficient funds to be able to do it ourselves, but that's a real practical problem. Is you can't take a piece of software and run it on your laptop and produce something wonderful. Those systems are just the training effort is mind-blowing. So I'm sorry, as I say, it's not my area, but
Okay. Well, I think it was curious to consider that in the conversation, because there are things that are evolving even quicker than we can even cope. I mean, we can keep up with this evolution because industry has virtually unlimited resources nowadays, and universities are trying to chase and to follow what's happening, and so it's difficult to.
It's not industry in general. We're talking about maybe three companies in the world that can do it.
Right. Yeah, exactly. Well, it's interesting to think about that and to remind ourselves that we can still do something useful in our scale, in our dimension, which is can still have a lot of value compared to what we found on the market. I have a question about when you mentioned the program correctness, right? Because we try to always keep the correctness of the code as a primary goal for our evolution. And so, of course, we have a very deterministic way of saying that this program is correct, because we have tests and we have use cases and we want to see whether the program goes through them in a correct way. Do you think there can be other ways of determining the correctness of a program, of a piece of code?
Well, so one of the nice things about working with bioinformatics, working with medical data, is the practitioners know their data is noisy. They know that no matter how, quote, perfect the software is, if you feed noisy data in, you cannot expect perfect answers. And they're very accepting of that argument. So what they want is an answer which is good enough now. They don't want a perfect answer in 6 weeks' time.
Right.
So they're very attuned to the idea of trade-offs, and that was one of the things that we exploited in particularly in Bowtie 2, was to get a trade-off where we trade accuracy against speed. And in that particular example, we won on both, but often in a multi-objective setting, evolution is very good at juggling multiple objectives in a way that's hard for people to do. So if you, and also you can produce lots of different answers. So although
that's very interesting. Yeah.
Although you may start off saying, I want to hit this level of performance, if the system shows you that there's another variant nearby which is twice as fast and not much worse, you might well be able to accept that. Whereas if that means you have to sit down and code for another 2 weeks, people would probably not be interested. So the ability to have this trade-off, even let me see if I get back that example. So obviously a lot of these examples here, the grey dots, they're doing considerably worse. Many of them are actually doing faster. But if you know, supposing you were at this point B, but then you run your evolution and it tells you that there's this point C available, it may not be immediately interesting for your application now, but supposing, as I say, the requirements for the shading, say you want to have 10 soldiers instead of 1, then suddenly the point C becomes interesting. And even if you demand to code it yourself, knowing that point exists is beneficial.
Right, it's a possibility that exists, and then you may want to consider eventually if the conditions allow that. Right. Okay.
And in terms of formal correctness, there has been a little work on transformations which are provably program equivalent. And so in principle, and it has been done to some extent, you can string together transformations which are have guarantees associated with them. Typically, people actually relax the guarantee and say, under circumstances we care about, these things were equivalent. So sometimes even the formal approaches get slightly fuzzy around the edges. So the mutations I've talked about have been essentially what seems like a good idea, a reasonable thing to try, but there have been work on trying transformations which are in some sense guaranteed.
Right. Okay. Okay.
And as I say, typically the guarantee gets a little fuzzy, and you start doing things like saying, well, in this particular circumstance, we know our inputs are, say, the positive integers, therefore we can do something or other, which might not hold for all possible inputs.
Okay.
But it's guaranteed for the inputs we care about.
Okay. Okay. Thanks. We have a question in the chat from Joan. He says, thank you for an interesting presentation. Do you know how of potential improvements to genetic algorithms by incorporating other evolutionary aspects, such as multiple populations, simulated environmental effects, etc.?
Okay. So a short answer is yes, there are a huge number of variations that have been tried. One of the nice things about genetic algorithms in evolution in general is you can spread your individuals across parallel hardware. So if you've got a machine with multiple cores, or you've got a cluster with multiple nodes, it becomes very easy to build a GA which makes very good use of the compute hardware by spreading the fitness evaluation out. So people have built parallel GAs which in the parallel computing community are known as embarrassingly parallel, because in the parallel computing environment, the getting an n-fold speed up from n computers is typically hard, whereas with something like GA, typically you can get very close to n-fold speed up from n computers with almost no effort. There's many variants on local search that have been incorporated into GAs and vice versa. I think the answer is yes. There's lots of things that have been tried, and in many cases they've been successful in terms of either speeding search or getting better answers.
Okay, thank you. This actually is a perfect segue for my for another question that I have. John, is it clear for you? Do you want some more explanation, or have your question been answered?
No, that was perfectly clear. Thank you.
Oh,
Thank you so much.
brilliant, brilliant, great. So, last question, I would say, so then we can go have lunch. Thinking about the parallelization and the parallel computing approach for these things. So I developed a symbolic regression algorithm, a Python code to evolve mathematical expressions to be used as predictive models for data science. And I developed a federated learning version of that. Are you familiar with federated learning?
It's not my specialty, no. Sorry.
Well, so the concept is to distribute the training process in multiple nodes. Especially this is useful in healthcare because you can keep the data stored in different hospitals. You send the model to be trained in the center of the data, and then you gather the model trained without moving the data. So it keeps the data privacy, but you can train machine learning models with the distributed data. Does it make sense? So only the model travel between hospitals and not the data. Okay.
And the goal really is to keep the data anonymous.
Yeah, anonymous, and to not move the data. So you keep the data in the firewall, the hospital. You put the model, you send the model to all the hospitals. They train inside their firewall, and you get the model trained back. And then you merge all the model together to get what would be the equivalent of the model you would have trained with all data gathered together. Okay. So this is fit basically is a distributed approach, parallel in that matter. And one of the things that I'm trying to understand, because it's a very new thing, symbolic regression in a federated way, is how to aggregate the models that are generated. Okay, so I have these trees that are mathematical expressions, as you showed before, and they are the result of the training of multiple hospitals. Let's say each hospital produce its own formula. We don't have any knowledge about what is the structure of the formula. We don't have constraints. They can contain any kind of expression, any kind of operation, etc. So I am in the problem of trying to merge those models. So when you have a deep learning network, you just put everything together, you average the weights of the neurons, and you're done in the most simplistic way. But this is different because you don't have any guarantees that the structure of the tree is the same. How do you merge the results coming from different training in one unique solution, if that makes sense?
So again, I don't know the answer, but I can suggest some things. So one of the things I have worked on is the idea of ensemble learning. So the classic examples are things like bagging and boosting, where it might actually be very useful to you, is that if you can take models that are built on particularly on different data, so you have diverse data, diverse models. What's been shown is you can combine those models In my case, I combine them using genetic programming, but there are a suite of techniques for combining models produced by other techniques or indeed by genetic programming, but generally diverse models to get a composite which is actually potentially considerably better than any one of the models by themselves. So as I say, probably the leading technique in that area is still the ideas of bagging and boosting, but essentially it's a machine learning technique which builds a single prediction from individual models and can do much better than those models if those models are themselves different from each other.
Right.
And potentially, in the case of hospitals, maybe they have different set of techniques. They may actually naturally produce diverse models.
Okay. I will look into that. It's something that is really new in the symbolic regression field. I've seen only one paper of last year proposing a federated aggregation algorithm for that, and so it's worth exploring because it's very, very new, and I'm doing my PhD on that basically.
So as you said, I would be tempted to look up the literature on bagging and boosting, and I'm trying to think. There was a generic term such as multiple classifier systems, which I think is the problem with hunting for the literature is if their names change, you may find that there's a very similar idea, but it was actually given a different name.
Okay.
So those three terms might well be useful. Certainly, we've been back in about 2000, 2001. The ideas of composite learning were very prevalent.
Okay, thank you. I will look into that, absolutely. So it's 3 past. If you have a couple of minutes to answer maybe the last question by Linglong.
Certainly, yes, sure.
Who has his hand raised, Linglong?
Yeah, hello. Can you hear me?
Yes, we can.
Oh, thank you. I was also curious about the problem you've discussed before. I mean, the further learning for different module on different location. From my side, I would focus on the data in different hospital. I mean, even the module itself is structured the same, the hyperparameters are all the same, but maybe the data itself, the data distribution will completely different or partially different, and even they have some shift or sort of problem for data itself. So maybe that's another kind of a challenge in this field. I mean, to aggregate those things without any data leakage. So I was wondering, because I'm also focusing on the symbolic area, and I think that could be a potential solution because the symbolic things could give us more insights beyond those hyperparameters weights trained by neural network and trying to guide us or give us some knowledge from the data itself. And then that will help us to think about how to aggregate the module or even the neural network from different hospitals, even different countries. But I haven't do a very detailed reviewing in this area, so I'm very curious about this part.
So one of the feelings I have from medicine, bioinformatics in general, is the practitioners tend to be very interested in the components of the model. So things like symbolic regression, genetic programming, as a practitioner, we always try to get the most accurate model. But in terms of the customer, the user, I think it's one of these things is you want to show people the model. Which one of the great advantages we have over neural nets is producing symbolic expressions. And what I would suggest is you make a model, you put it on a slide in front of the people, and you use their terminology. You don't call things X1 to XN, you use their names.
Right.
And the example I have in mind is I did some work with GlaxoSmithKline on looking at P450 and the interaction between P450 and various chemicals. They put up a blind trial. I forget the numbers, but it was something in the order of 10,000 molecules. And they had a 2-day event where 12 different approaches were presented. We turned up late on the second day, and these chemical guys had 2 days of people talking to them about random forests, about neural networks, about SVMs. And it was approaching 5. They just wanted to go home. And so I get the last slot and I start talking about GP, and you can see they're all looking at their watches until I put up the slide with the components that they understood. They had 2 days of black boxes. And here was somebody who was actually presenting chemistry to them. And the meeting went from totally bored, looking at their watches, to, gee, wow, I told you this sucker was important 2 years ago, and you didn't pay any attention. Put stuff on the slide that they understand. That was much more important than the fact that we won the competition. The accuracy didn't matter. In practice, what they cared about was what was in the model.
Yeah.
And if they understand the model, they can do something with it.
Right.
If it's black box, well, yeah, okay. But what they really want to do is understand the science. And a symbolic model may well help them. Use the names that they use.
Yes, I see.
Very precious advice. Very, very useful. Yeah.
Put your model on a slide, present it to them.
Yes.
Even if it's a rubbish model, if they understand how it works, that's much more important than the fact it's not all that predictive.
Yeah, exactly. That's also kind of things we are currently working on. We're trying to transform those numerical value or lots of complex data into a symbolic, and it's much more easier for people to understand what we are trying to do and what we find. That's a very good good method. That's like. Thank you so much.
Okay.
Thank you indeed. Listen, I would say thank you again for joining us, for giving your time to speak to us, and I would call it a day. And I don't know. Let's catch up when we have another genetic programming papers to discuss.
Okay. And David has got copies of the slides if anybody wants them.
Right, I can share with anybody here in the chat if you want. Yeah.
Yeah. Thank you so much.
Thank you very much, and I'll see you next time for the next seminar. Bye.